I'm going to be presenting today on the Ribosome Labs round one results. Some of my methods and how I developed my design for the round one. The ribosome, the molecular machine that turns RNA codons into proteins that performs important functions to our bodies depending on how they are folded. Millions of ribosomes fit in each one of our cells. They're very small. In this lab, we are re-engineering the ribosome for medical purposes, as well as enabling scientists and companies to make state-of-the-art materials for such things as LCD screens to waterproof materials. And the applications that could come from this are really limitless. The ribosome subunits. In this lab, we're working on the ribosome's main subunits 16S and 23S. And I'm going to be talking mostly about the 16S subunit. Let me open up the, this is a graph of the results from round one. It's pretty straightforward. This one was at the optimal ISAT conditions. And what it is this wild type right here produces so much GFP proteins. And it's at one, our goal is to beat that, to re-engineer it to perform better. As you can see, many of these, 4 or 5, beat the ribosome in round one. This is mine here. It performed 22% better than the wild type. As you can see, we did much better than the pilot round here, where Eli Fisker had the best one with about the same output of GFP. I'm going to open a document here that was made by Eli Fisker and Digital Embrace. It's really great for learning the round one results so that you can jump in on the next round and be able to make good designs by analyzing these results. It also has notes for each design, how many mutations it has, if it had any motif violations or protein violations, and other data also. The best designs are at the top with the dark red, and you can also jump to the designs with the link there. So I'll just open up my design here. And what I want to do is go around and show you some moves that I did and what they affect. So here's a move here. If you look up here at the natural target delta, it's at 173.9. Now, if I take this mutation away, it goes up, and what we want is lower target delta. That means it's strengthening the ribosome subunit and could perform better because of it. So that's one move. Another move here does pretty much the same thing. So this was my method on trying to make a good design. Now this one here, 48 to U. I also had this move in my pilot round design that didn't do as well. I think that it didn't do as well because I had other mutations that weren't good mutations. They violated proteins and motifs. And I'll talk a little bit more about that just in a bit. But you can see here that this also lowers the delta. Now, out of all my moves, they all did that. But this one in particular, I think, is very interesting. 853. That was originally the wild type was the GU there. So that's like 1, 2, 3, 4 GUs in a row. So when I mutated it, the delta actually goes up. The main thing about this is look at the picture here. You can see the red, that's misfolding areas. Now, without that, I'm going to click on it again and look up at the red that disappears. You can see that it really helps this turn white. Let's see. So all 6 of my moves, they were all very safe moves with no protein violations. Now, let me show you about the protein violations and motifs.
Henry Dennis, he says, I used 853 with other bases in a way that lowered the delta in whitened areas. Why? Why did it do that?
It did that because, let me just put this back to where it was and click here. You can see that it's flashing red. And when I mutated it, it strengthened it. And by strengthening that, it will lower the target delta. I also thought for it that it would be a good move because of all these GCs in a row, and I kind of wanted to interrupt that. This here is the document that I think Raju and Eli Fisker put together to let us know which bases are violating motifs. Motifs are sequences that are proved to be good for certain structures. And the 16S subunit has many motifs that the wild type uses. Eli posted something in the forum after the pilot round about these violations. And he was thinking it might be good not to violate them. So you can like look up a base here and you can see this one, base 3, has a minor motif violation. The motif violation, they go from here to here. If you got a Y there, it'll show you which motif you're violating. And then on this side, from this to over here, are the protein violations. Proteins help the RNA and the subunits fold certain ways and help them to do their functions. So the thinking is when you violate that, it could hurt your design. So every one of my moves and my winning design doesn't have any protein violation or motif violations. The best way to get these super safe moves, super conservative moves, I need to go to the bottom here. They're at the bottom right, and here they are. What you do is you just copy that. You paste it into Notepad or whatever. Then save it. Let's see. Yeah. Super safe mutations. Um, and then you'll have them here where you save them. Uh, so in my design, I had every one of my mutations are in this list. And I think it's a good idea to do that. Let's see. Let me just paste this into the chat. Well, I guess I'll do that afterwards. I'm having technical difficulties. Okay. So I want to let you know that I came up with this method by studying the palette round. Here's the palette round here. And here's my design. It doesn't do as well as the wild type. And that's because I do have that 48U in there, but it wasn't able to do so well because I had the protein violations and the other violations. So I think if this one didn't have the 48U, we wouldn't even have done that well. And I also want to say that these designs down here that they didn't do as well are also very good tests and good data for us, just because they tell us what not to do and other data, stuff like that. In round 2, what I did was one of my designs, I took this move out. I just want to see how important it was. Is the design going to do worse? Is it going to do better? And I also want to say that there's also very many other methods that people are doing. They're testing things like trying a different motif, different tests that will give us a lot of insight on the ribosome.
First of all, great presentation. Thank you. Thank you for putting that together. That was eye-opening for me. I had 2 questions. One was this showing the safe mutations, showing the residues, the bases that are involved in motifs, showing the bases involved in proteins. I mean, how are you doing? You're doing that manually by looking at the spreadsheet now, or would it be useful to provide some kind of booster that, I don't know, lets you put up black marks or something on those residues?
Yeah, you can look up each base. You can look up each base going down here and then finding it and then looking to see if it has it. The easy way is to copy the super safe moves, which none of them have violations.
Mm-hmm. Mm-hmm.
And what you can do is you can pick them out and then go to the puzzle and try them out, see what the delta does, see how it fixes mismatches, and you know that the moves are going to be safe.
Mm-hmm.
And it's just a matter of picking the right ones to make a good design. Like sometimes you can have 2 or 3 good moves, but together they really don't work. So it's just finding the right concoction pretty much.
I'm just wondering if we could have a booster that, maybe puts black marks on all the nucleotides that are not safe. So this is the blacklist. And then I feel like that should be a pretty booster to make.
I think it would be.
You could turn on and off, so turn on temporarily. Okay. Oh, I see. So there's a comment from Roger. that Eli, Digital Embrace, Andy, and Omri are working on the booster to flag protein contact sites and motifs. Okay, that's good. I have to say, I'm elated that those lists of bases came in handy. I was talking to Eli about them mostly, and then I'm so glad that it's influenced your work, which has been yours and others' works, because it's been so successful. I had another question. It's kind of about, first of all, you made this point that even the failures, the ribosomes that don't work, are powerful data points. And so that's great feedback to have. Now, on vaccines, we've discovered ways to synthesize now RNA molecules that are maybe up to 1,000 bases long and arbitrary sequences. So that's pretty awesome. We couldn't do that before February. And we can get a lot of those, okay? And so one thing that the developers are trying to understand is, like, for example, for the next rounds of the ribosome puzzles, how valuable is it to provide, okay, so with those technologies, we could take like a subset of the 16S ribosome or a subset of the 23S ribosome, like right around the active site, and allow players to make not just 8 or 16 solutions, but maybe 96 solutions. So expand the synthesis scale for those shorter subsets. And we can in principle, we get data turned around faster. So one question then is, can you imagine that those would be a useful set of tests? You and others. I just saw a note from Stephen Clark. That would be incredible. So, and if so, what subregions would be most valuable? Like, are there particular problem areas where you'd like to be able to generate 96 hypotheses, but those areas have to be close to each other within the sequence? I just want to throw it out there. If anything came to mind, it's like, oh, I really wish I could go to town on this particular subset of the ribosome.
I would like to do the ones that are close to my mutations.
Yeah, each of those. Yeah.
See if we can get it better, do it better.
Yeah. Okay.
And I think that would be a great idea to make a booster that would notify us when we do violate those. Like some people are doing tests that want to violate it and they want to see.
Yeah, right. Mm-hmm.
But for those that are wanting to just go that way, that method, a booster like that would be very, very valuable. And the thing you're talking about, that would be great also, to be able to test all these different areas like that.
Okay, so that's something that we can talk about. Especially we'll talk about tomorrow with Antje is the lead experimentalist in the ribosomes, and we're hoping to have a really fun discussion tomorrow.
Great, looking forward to it.
Yeah, me too.
Amy Barish, she's asking, how did you originally choose your moves?
Just when, at the beginning of my presentation, I'd go around and see what lowered the delta and fixed misfodes. That was the pilot round. Now in the first round where I did my winning design, I was taking out protein violations and motif violations. That's how I picked them. So they would have to lower energy, lower delta, fix misfodes, and not be connected to any proteins or motifs.
Henry Dennis, would a list of the effect of each mutation at the default delta of the 16S be helpful?
Oh, yes. Document, if someone did a document like that, I would definitely be using it. It would be very helpful.